All right, we're going to get started. Like I said, talking about DNA. This is something you went over in freshman biology when you went over it, especially if you were in my class. I know that you guys went over it, but all freshman biology classes go over the structure of DNA and how it acts. The importance of DNA. In 2009, the swine flu outbreak, scientists discovered it was a mutant strain that originated through a combination of viruses from birds, swine, and people. Now, since then, we've dealt with the avian flu, we've dealt with the West Nile virus, we've dealt with Ebola, and now we're dealing with the coronavirus. This is very relevant. DNA is very relevant to today and our problems today, and the vaccine that people are making and getting. So all of these are very simple molecules. They're nucleic acids and a little bit of protein. And to fight them, we have to know what they're made of and how they work. Good morning, Daniela. So DNA was known to be a chemical in cells by the late 1800s. By the 1930s, experiments convinced scientists that there was only one molecule responsible for heredity rather than a mixture. They used to think it was just proteins, and then they discovered the nucleic acid. In the 1940s, scientists knew that chromosomes carried genes and are made of DNA and protein. In the 50s, a series of discoveries convinced the world that DNA was the molecule that is responsible for inherited traits. Protein is so important in our bodies that a lot of scientists thought, oh, it had to be protein that was controlling our traits, but they were wrong. They did their scientific method where they did some hypothesis, did some tests, realized they were wrong, and it was the nucleic acids every single time. The discovery of DNA as the hereditary material ushered in a new field called molecular biology. That is the study of heredity at the molecular level. What kind of chemicals is it made of? How do these chemicals behave? How are they shaped? How do they interact with each other and other molecules in our bodies? In the 1950s, scientists knew the atoms that made up DNA. They also knew how they were bonded, but they did not know how all of these molecules were arranged. We're going to watch a short video.
The discovery of the structure of DNA was one of the most important scientific achievements in the last century, in human history, in fact. The now famous double helix is almost synonymous with Watson and Crick, two of the scientists who won the Nobel Prize for figuring it out. But there's another name you may know too, Rosalind Franklin. You may have heard that her data supported Watson and Crick's brilliant idea, or that she was a plain-dressing, belligerent scientist, which is how Watson actually described her in The Double Helix. But thanks to Franklin's biographers, who investigated her life and interviewed many people close to her, we now know that that account is far from true, and her scientific contributions have been vastly underplayed. Let's hear the real story. Rosalind Elsie Franklin was born in London in 1920. She wanted to be a scientist ever since she was a teenager, which wasn't a common or easy career path for girls at that time. But she excelled at science anyway. She won a scholarship to Cambridge to study chemistry, where she earned her PhD. And she later conducted research on the structure of coal that led to better gas masks for the British during World War II. In 1951, she joined King's College to use X-ray techniques to study the structure of DNA, then one of the hottest topics in science. Franklin upgraded the X-ray lab and got to work, shining high-energy X-rays on tiny, wet crystals of DNA. But the academic culture at the time wasn't very friendly to women, and Franklin was isolated from her colleagues. She clashed with Maurice Wilkins, a lab mate who assumed Franklin had been hired as his assistant. But Franklin kept working, and in 1952, she obtained Photo 51, the most famous X-ray image of DNA. Just getting the image took 100 hours. The calculations necessary to analyze it would take a year. Meanwhile, the American biologist James Watson and the British physicist Francis Crick were also working on finding DNA's structure. Without Franklin's knowledge, Wilkins took photo 51 and showed it to Watson and Crick. Instead of calculating the exact position of every atom, they did a quick analysis of Franklin's data and used that to build a few potential structures. Eventually, they arrived at the right one. DNA is made of two helicoidal strands, one opposite the other, with bases at the center like rungs of a ladder. Watson and Crick published their model in April 1953. Meanwhile, Franklin had finished her calculations, come to the same conclusion, and submitted her own manuscript. The journal published the manuscripts together, but put Franklin's last, making it look like her experiments just confirmed Watson and Crick's breakthrough, instead of inspiring it. But Franklin had already stopped working on DNA and died of cancer in 1958, never knowing that Watson and Crick had seen her photographs. Watson, Crick, and Wilkins won the Nobel Prize in 1962 for their work on DNA. It's often said that Franklin would have been recognized by a Nobel Prize if only they could be awarded posthumously. And in fact, it's possible she could have won twice. Her work on the structure of viruses led to a Nobel for a colleague in 1982. It's time to tell the story of a brave woman who fought sexism in science and whose work revolutionized medicine, biology, and agriculture. It's time to honor Rosalind L.C. Franklin, the unsung mother of the double helix.
So that's a little background on how we came to the structure of the double helix. So DNA and RNA are both nucleic acids. They're one of the four main organic molecules you studied previously. We talked about carbohydrates, proteins, lipids, and we talked a little bit about nucleic acids, but that was only because I knew we were going to have a whole chapter later to talk about it. But they consist of chemical units called nucleotides. Nucleotides are made of sugar, phosphate, and a nitrogenous base, which is just a base made of nitrogen. Nucleotides are joined by covalent bonds between the sugar of one nucleotide and the phosphate of the next, forming a sugar-phosphate backbone. You can see that arrangement in the picture on the right. The sugar is the pink molecule, and it's attached to a phosphate, and it goes that way all the way up. There are four different bases which make each nucleotide different. Two of them are called pyrimidines, and they are thymine and cytosine. 99% of the time, they just go by their letters. We hardly ever call them thymine and cytosine. We just call them T and C. And they are a single ring structure, which I know the picture looks like a hexagon, but we just call that a ring in organic chemistry. And then the other two are called purines. They have a double ring structure, just two rings stuck together. And those two are adenine and guanine, A and G. And when these nucleotides pair together like they will, a pyrimidine is always paired with a purine. Here's some differences, main differences between DNA and RNA. In DNA, your sugar is deoxyribose. That's where the D comes from. In RNA, the sugar is called ribose. That's where the R comes from. They each have four bases. DNA has the A, C, G, and T, like we just talked about. RNA, however, has A, C, G, and U. The U stands for uracil. DNA is double-stranded, obviously, and then RNA only has a single strand. We'll talk about the purpose of RNA here in a few minutes. The model of DNA is like a ladder twisted into a spiral. The ropes at the sides, the parts you would kind of hold on to, are the sugar phosphate backbones, held together with covalent bonds. And then each of the rungs in the middle represents a pair of bases connected by hydrogen bonds. The rungs are the part you would step on if you were climbing up a ladder. Base pairing rules. Erwin Chargaff was a scientist who discovered how the base is paired in the rung structure. And they pair in a complementary fashion. Complementary is just something that goes together. Peanut butter and jelly go together. They're complementary flavors. So are peanut butter and chocolate. Adenine, A, always pairs with T. And then cytosine always pairs with guanine. C's and G's always stick together. And if you need an easy way to remember that, A and T are made with straight lines. They're made with sticks, A, T. And then C and G are the curly letters, and they always stick together. You're going to need to know this for your quizzes and tests this week. Like I said before, purines always pair with a pyrimidine. So you have an even structure all the way down. And the number of A and T molecules are always equal. So if you have 5 A's, you're going to have 5 T's. And same with C's and G's. If you have 10 C's, you're going to find 10 G's. So really quickly, you're going to see, these are short versions of the questions you'll see, but I have a poll for this one. Wrong one. Don't answer that one yet. This one, the complementary DNA. So what is complementary to the DNA strand CTC? You got 3 letters. What pairs with a C? What pairs with a T? What pairs with the last C? What is complementary to the DNA strand AAA? If you see 3 A's, what's going to be on the other DNA strand? And then the third one, what is complementary to the DNA strand GGT? What's going to pair with the 2 G's? What is going to pair with the T? There are rules, and the rules are right there on that slide. The letters with the straight lines stick together. The curly letters stick together. I'll give you guys about 15 more seconds to answer. Like I said, these are the type of questions that are going to be on your test this week. Good, yep. CTC pairs with GAG. AAA pairs with 3 T's. And GGT pairs with CCA. So those of you that answered, you guys all got these correct. Very good. All right, DNA replication. So there's 3 processes to the whole protein synthesis. DNA replication is when a cell reproduces, a complete copy of the DNA must pass from one generation to the next. DNA replication ensures that all the body cells in multicellular organisms carry the same genetic information. And Watson and Crick's model suggested that each DNA strand serves as a mold or template to guide reproduction of another strand. So we talked about the cell cycle a couple of chapters ago, maybe last week. We talked about G1, M, G2, S, or G1, S, G2, and M are the 4 phases of the cell cycle. This is what happens during the S phase, the DNA synthesis phase. Right before a cell goes through mitosis and splits in half. All the DNA has to double itself so that the 2 daughter cells get the complete set of DNA. So this is, in general, how DNA replication happens. You can see the parent strand is blue. It splits, and then daughter strands, in orange, get made from that parental strand. And I like to split this into 3 steps to break it down and make it a little easier to remember, hopefully. So DNA replication in eukaryotes, in us, begins at specific sites on a double helix called the origins of replication. It proceeds in both directions, making replication bubbles along the DNA strand. One side, the side basically going forward, replicates continuously. That's the top strand in the little GIF. The other side, which would be the bottom strand, has to proceed backwards. So it makes fragments and then glues those fragments together. And that was discovered by a scientist named Okazaki, so we named the fragments after him. So step 1, enzymes unzip or unwind the double helix to make a replication bubble. Helicase is the enzyme that is responsible for that. Helicase rips open the double helix. And then each strand acts as a template or hard copy. If you've ever gone to the copy machine and made copies, you know you have that one piece of paper that you stick in the copy machine and you push a button and a bunch of copies come out. That original paper is called a template. So the original information from the DNA that gets copied is called the template. Step 2, bases that are floating free in the nucleus bond to their mates on the original DNA strand. For example, if the parent strand has a T, the daughter strand is going to have an A to pair with it because the T's and the A's and the T's stick together. If the parent strand has a C, the other curly letter, the G, will also stick to it. And these are simplified GIFs of what is happening during this step. Step 3, enzymes covalently bond the 2 new molecules of DNA together. So like we said, the sugar and the phosphate, sugar, phosphate, all the way up the molecule, need to be covalently bonded. And DNA polymerase is the only enzyme needed to bond the continuous strand. But then you also have ligase that bonds the Okazaki fragments together. Each new DNA has 1 old strand and 1 new strand in its double helix. It's called a semi-conservative process. Semi means part or half. We talked about that when we talked about semi-permeable membrane, when we talked about cells. So DNA is the template, but enzymes do the actual work. DNA just kind of lays there and lets itself get replicated. But this process is called semi-conservative because each new DNA molecule has half old information and half new. It conserves half of the information, so it doesn't have to start over from scratch, because that would use a lot of energy that we just don't need to use. So normally we would do a worksheet in class, but we're just going to do this all together. So make sure DNA is always split into groups of 3. Write down the complementary base pairs below based on Chargaff's rules. So you guys tell me, what pairs with that first A? What letter? We're going to do this together by the chat box. If we were in class doing this, what would you write down to pair with that A? Very good, Mike. T, what pairs with the T? A, and then what pairs with the G? C, good, Julian. Good, Allison. All right, let's take them by 3s now. What's going to pair with CTA? GAT. Good, Julian. Next one, what pairs with GAC? CTG. Good, Mike. What about AAA? That was one in our poll. TTT. Good, Julian. Good, Sierra. What about CCG? GGC. Good, Mike. Good, Julian. And then last, TAA. ATT. Very good. We'll be doing this a couple times throughout the notes. Like I said, if you were sitting here, I would have given you a worksheet and we would have been working this out together. But complementary DNA strand, there it is. This is the kind of stuff you'll be doing in your test and your quiz this week. All right, genotype determines phenotype. So an organism's genotype is its genetic makeup, which comes from the sequence of nucleotide bases in DNA. And then the phenotype is the organism's physical traits. They are coded for by the genotype. So we went over this last week. This should be review. The genotype is the letters heterozygous, homozygous, recessive, dominant, big A, small b, or one of each, big S, small s. And each of those codes for an actual physical trait of an organism. The central dogma of biology states that information flows in one direction from DNA that gets transcribed into RNA and then translated into polypeptides or proteins. And then proteins actually code for phenotypes or physical traits. So this whole process is called protein synthesis, the making of proteins. And there's 3 main processes that happen during the central dogma. So like I said, and information flows from DNA to RNA to proteins. When DNA copies itself, we just went over, it's called replication. When DNA gets transcribed into RNA, it's called transcription. And then when RNA gets translated into a protein, it is called translation. RNA is the middleman between DNA and proteins. So we went over the structural differences between DNA and RNA. Now we're going to go over the purpose of RNA. Transcription. Again, all these processes have 3 steps, or I've broken them down into 3 steps to make it easier to visualize. So RNA polymerase binds to a promoter or start site to initiate the process of transcription. And this step actually has a name, just called initiation. When you initiate something, you start it. So this is the start of transcription. So RNA polymerase has to find the start site and bind to it to be able to start this process. Step 2 is called RNA elongation, or just elongation. Free bases pair with one strand of DNA, not both. And the rules are a little different. C's and G's, the curly letters still stick together. But remember how we talked about RNA has U's. So you will still see T's in the DNA. When you see a T in the DNA, it will pair with an A. However, if you see an A, it will now pair with a U when RNA is being made. So if the question asks for an RNA molecule, there will be no more T's. There will only be U's. So you have to make sure you pay attention to what the question is asking in your quiz and your test this week. Is it asking for DNA or is it asking for RNA? The RNA strand eventually peels away from the DNA template as it gets longer. RNA polymerase bonds the nucleotides and the mRNA strand together. And then step 3 is called termination. RNA polymerase reaches a sequence of base pairs called the terminator sequence, which means stop. The mRNA strand separates from the DNA, moves out of the nucleus through the little nuclear pores all around it, into the cytoplasm to find a ribosome for the next step, which is translation. And then the DNA just acts like nothing happened and rewinds. You can see in the picture, the DNA is just rewinding behind it. Once the RNA polymerase comes to the terminator sequence, everything stops. The RNA just separates and moves along. Codons. Experiments have verified the flow of information from gene to protein is based on a triplet code. Codons are nitrogen bases grouped into threes on the mRNA strand, and then each codon gets translated into a different amino acid. All right, next part of the worksheet is transcription. Open your chat box and tell me, ATG, what are the complementary bases now using our new rules to ATG? What does the A now pair with? What do we got? Using our new rules in our elongation slide, you guys should have just written down. What does the A now pair with? What is ATG going to look like when you are making an RNA strand, an mRNA strand? Good morning. No, nobody wants to.
Isn't it is it you supposed to replace the T in the strand, right?
Right. So what does A pair with now?
U instead of T.
There
So
you
it'd be, yeah, UAC.
UAC. There you go. Good, Allison. Good, Shay. What about the next three? What is CTA going to pair with?
GAU.
AU. Yep. And then GAC is going to pair with
CUG.
UG. And then AAA pairs with, good, Julianne.
UUU.
UUU. And then what is CCG? Good, Sierra. Good, Marcos. GGC. And then last three, what does TAA pair with? Good, Julian, Mike, Samantha, Marcos. Good, everybody's getting the hang of it. All right, so there's your DNA strand. Now when I ask for an RNA strand, this is what you see. You don't see any T's in the answer. It's all U's. All right, now we move on to translation. And there are different molecules you have to know before we get into translation. So translation is the conversion from the nucleic acid language, the A, C, G, T, U, to the protein language. And translation requires many different molecules. The mRNA strand that we just made during transcription requires ATP. Remember, that's our energy molecule. Various enzymes, just like we were talking about, helicase, ligase, polymerase, all that. So these are new enzymes. Ribosomes. We learned way back in chapter 4, I think, that ribosomes are the site of protein synthesis. This is where proteins are actually made. And then a new RNA molecule called transfer RNA, or tRNA. We'll talk about the structure of that in a minute. So it looks like, to me, it looks like a little bent over old man. So each tRNA carries an amino acid on top. That would be his little face. And then an anticodon on the bottom, his 3 little feet. So on the mRNA, we separated them all into 3s and we called them codons. On a tRNA molecule, at the bottom, you have 3 bases. Those are called anticodons. The codons on the mRNA strand attracts complementary bases, just like the other 2 processes we talk about. And the complementary bases are now called anticodons, and they are found on a tRNA. So step 1 is also called initiation of translation. The mRNA binds to a small ribosomal subunit. A special initiator tRNA binds to the start codon on the mRNA where translation will begin. And the codon AUG is always the start codon. That is always the group of 3 that translation will start at. The initiator tRNA is the complement AUC that carries the amino acid methionine. Usually abbreviate amino acids, so we just call it MET. And then the large ribosomal subunit binds to the small one. And there's different sites on that ribosome. And the tRNA fits to the P site on the large subunit. I don't know why this GIF doesn't start over and over, but I can escape and then present it again. Oh no, it won't. There it is. So there's the small subunit. There's the tRNA molecule. There's the large subunit. And that's called the initiation complex, and it's the end of step 1. And then elongation. The tRNA and the mRNA come together in the ribosome according to their base pairs. The amino acid bonds to each other on top of the tRNA. And the result will be a long chain of amino acids which make up a protein. And here it is kind of fast forwarded. But then you have termination. Elongation continues until a stop codon reaches the ribosome's A site. The completed polypeptide is free and the ribosome splits back into its subunits. If we want to see that process again. You can see on top is the polypeptide chain, and eventually everything just goes its own way. Translation, we had our mRNA codons. So this is our mRNA strand. We already did that. We're going to copy it and paste it down here. And now we're going to list the tRNA anticodons. So what goes with UAC? It's still RNA, so I still shouldn't see any T's. What goes with UAC? AUG, good. What about GAU? What's next? What goes with GAU? What pairs with G? What pairs with A? What pairs with U? CUA. Good, Sierra, good, Mike. For the next one, CUG.
GAC.
Yeah. And then the next one, UUU.
AAA.
Good, Sierra, Mike, Julian, Shay. What about GGC?
CCG.
CJ, good. Marcos, Julian, Sierra, Shay. And what about AUU?
UAA.
Good. There you go. So there's your tRNA anticodons that pair with the mRNA codons. All right, so overview of the whole thing. DNA to RNA to protein. As it is made of polypeptide, coils and folds and assumes a three-dimensional shape. There's another chapter we'll be talking about. This we already talked about it, I'm sorry. We talked about the primary structure, is the order of the amino acids. The secondary structure gets folded into an alpha helix or a beta pleated sheet. The tertiary structure folds on itself even more. And depending on the attributes or the characteristics of the amino acids will depend on how they interact with each other. Are they going to attract? Are they going to repel? How are they going to make that 3D structure? So transcription and translation are how genes control the structures and activities of the cells. So genetic code. How do we know which codons and anticodons match which amino acid? We have to be able to read the code. This is a chart of all 20 amino acids found in living things. So we are a collection of 20 amino acids in different sequences and orders. That's it. That's all we have in us. So it's the order of these amino acids that determine which proteins are expressed. So for example, the protein for blue eyes is different than the protein for brown eyes. And those proteins are coded for by our individual DNA. The genetic code is the set of rules that convert a nucleotide sequence in RNA to an amino acid sequence. And like we said, there's based on a triplet code. We take them as groups of 3. And there's only 64 triplets that can be made from the A, G, C's, and U's. And out of the 64, 61 code for amino acids. It includes this start. Methionine is always the starting amino acid. And then 3 are stop codons, which are instructing the ribosomes to end the polypeptide chain. So we take them 3 by 3. We're doing AUC as an example. So the first letter of the codon is found on the left side of the table. So this actual chart is known as the genetic code. Sometimes you'll see it as a circle, but I like to teach you guys the chart. And it's really easy to read. So you can see down on the left, UC, AG are all listed. And our first base of our triplet code example is A. The A is the third one down, so now you have a row to look in. Our second letter is U. The second letter of the codon is found on the top table, on the top of the table. So now you have a row and then you have a column. And where that row and column meet, you have a box. That is the box you are looking in for your specific amino acid. And then the third letter is found on the right side of the table. So within that box, you have to look for where that third letter points to. So there's your AUC, and not all charts you'll see will have the triplet codons listed in the middle. If you blow up that square, you read the square where all three letters intersect. AUC equals ILE. I know it's a little blurry. That's what it says, which is the abbreviation for the amino acid isoleucine. So here's another chart with the directions on it. So using the chart below, you're going to figure out the amino acid sequence coded for by each RNA molecule. Again, you guys are going to be doing this in your quizzes and tests this week. So we took it 3 by 3. I'm going to give you the 3 letters of an RNA strand. It's the RNA strand we transcribed and translated. Use the chart that's right here in the notes to figure out which amino acid it codes for. So AUG. I will help you with the first one. You have your first base is A. So this is the row you're looking at. Second base is U. So you have your box where they meet, where they intersect. And then your third base on the right is G. So that G points to the bottom in the box. So there's your amino acid where all 3 of those letters intersect. Let's see if you guys can do the rest of them. What amino acid does CUA code for? And then GAC, AAA, CCG, and UAA. Oh, I don't know why I'm sharing it with you guys. You guys got to tell me. Relaunch. Continue. Can you guys see it?
Yeah, I can see it.
So like I said, these are going to be questions in your quizzes and tests this week, which is why we are practicing it. Are you guys answering? There we go. Give you guys another 30 seconds or so. All right, and there's the results. So you guys, or the 5 of you that answered, doing pretty well. AUG is methionine, CUA is leucine, LEU. GAC is asparagine, ASP, AAA is lysine, good. CCG is proline. I don't know who chose valine, but it is proline, so make sure you review that one. And then UAA is stop. Very good. That is how you read the genetic code. I lost my notes. We are almost done. The interactive portion is done, so now I just get to share the rest of the notes. All right, so an amino acid chain equals a protein equals a polypeptide. These are all words that you will see, and they can kind of be used interchangeably. If you see the word protein, polypeptide, amino acid chain, they all refer to the same thing. So there's only 20 amino acids that determine all of our genes. Our genes are determined by the number and sequence of amino acids that are translated by the 3 processes: replication, transcription, translation. Are we going to end up with 5 amino acids? Are we going to end up with 100? Are we going to end up with 500? Just like the words of the alphabet. Some words in the alphabet, we only have 26 letters, but we have millions of different words in our English language alphabet. Some words are 2 letters long, some words are 10 letters long. And they're all made up of a different order of letters. So because diverse organisms share a common genetic code, it's possible to program one species to produce a protein from another species by transplanting DNA. And there are different classes in microbiology in college, if you ever take it, is one. Medical interventions here, if you ever take it, is another. We do this. But we insert a jellyfish gene into bacteria to make it glow. And you can see scientists have done this with several different animals and organisms, even mice and rabbits, which are mammals like us. A mutation is any change in this nucleotide sequence of DNA. Mutations can change the amino acids in a protein. They can involve large regions of a chromosome or just a single nucleotide pair, which is what happens with people who have sickle cell disease. Their DNA is just one letter off, and it causes all kinds of havoc. Types of mutations. A couple of general categories. Nucleotide substitutions is just the replacement of one base by another. That's what happens in sickle cell disease. Or there can be deletions or insertions. Those are exactly how they sound, where a nucleotide gets deleted or inserted in a place where it's not supposed to be. Insertions and deletions will change the entire reading frame of the genetic message. Like I said, we read these 3 by 3 by 3 by 3. If you eliminate one of the 3, the whole reading frame shifts. And I have an example that'll make more sense in a minute when you see it. But this can lead to disastrous effects. So this is just a cutesy little GIF. Insertion, you can see it gets inserted where it's not supposed to be. Deletion, a nucleotide gets deleted. And then a substitution is the T gets replaced with a C that's not supposed to be there, or a G. And then this is the practical example that you can see how it would affect an actual protein. So the fat cat ate the rat is a normal sentence. The words all make sense. They're all made of three lettered words. So each word represents an amino acid in a protein sequence. A substitution causes a point mutation. The F becomes an H. The hat cat ate the rat. It's still readable. It makes a little less sense, but you can still read that sentence. But deletions or duplications or insertions will cause the entire reading frame to shift one way or the other. So the first one is a deletion. You delete that F or that H altogether, the entire reading frame has to shift because you still have to read it as groups of three. And now the sentence makes absolutely no sense. So it's going to become a completely different protein in the end. And like I said, can lead to disastrous effects, different disorders and diseases.